Under the first subtopic, the process of digestion, third point is food in the small intestine. The small intestine is a 7-meter long coiled tube. It is divided into 3 parts. The upper part is duodenum, the middle part is jejunum, and the third part is ileum. What happens to the food in the duodenum we have discussed. Now, the food from the duodenum reaches the ileum through jejunum. It is because jejunum itself has the word meaning empty. When the jejunum area is cut open in the dead human body for any finding, then it is always found empty. So, jejunum only provides a passage for the food to move from the duodenum to the ileum. So, we are going to start with the ileum in detail. Ileum is the longest part of the small intestine. It is 4-meter long structure. It produces 4 kinds of enzymes: erepsin, maltase, sucrase and lactase. Erepsin acts on the peptones and peptides and convert them into amino acids. Maltase changes maltose into glucose, and sucrase changes sucrose into glucose and fructose. Lactase changes lactose into glucose and galactose. So, the product amino acids, glucose, fructose and galactose are the simplest form of the different kinds of nutrients which our body can absorb easily. That means here the process of digestion is completed. Our next subtopic is absorption of digested food. Here the process again is done in the ileum of the small intestine. The ileum walls have many finger-like projections known as villi. Every single pointed structure is known as villus, and together it is known as villi. In between the villi there are small circular hollow structures. Under them, in the lining of the ileum, small glands are present which secretes some juices, and that juices comes out from this hollow circular structure which helps in the process of digestion. Here I have drawn 2 diagrams to explain the structure of villi. It is said that the villi absorbs the digested food. So, I am here to explain how it is possible. This is the structure of villi. Inside each finger-like projection, I have drawn red and black color blood capillaries. These blood capillaries are thin, and the down one, which is in the lining of the small intestine, these blood capillaries are thick. So, these are representing the main blood stream, and this one is the blood capillaries present inside the finger-like projections known as villi. So, here is the diagram of microscopic structure of villus. In this, we will study in detail about it. Each villus has the inner, single and thick lining of epithelium cells. This black color, outermost inner lining, is showing the epithelium cells. Next to it, in the inner side, here is the red color line and here is the black color line. Just like this diagram, red one is showing the artery, black one is showing the veins, and in between these red lines are the interconnecting tissues known as capillaries. That means capillaries are present in between the arteries and veins. That means it connects arteries and veins. Then next to it is this inner part which is known as lacteal. All together these are known as blood capillaries or blood vessels. In common I have written here blood capillaries, and in detail they have got their names in this diagram. Now the end product of the digestion you have to remember here. First one is the simple sugar, that means glucose, fructose and galactose. Then from the protein we got amino acids. So the epithelium cells have the capability to absorb simple sugar and amino acids because they are having very small size molecules. So they absorb the sugar and amino acids and transfer into this blood capillaries. Then from this capillaries it passes into this thick one, main blood stream. Then remains the fatty acids and the glycerol. So they are absorbed by this lacteal area of the villus. They absorb fatty acids and glycerols, and again it enters into different ducts for different purposes which goes to different organs. In this way we have completed the absorption of digested food with the help of the diagram.